Scientists in China have created the first monkeys cloned by the same process that produced Dolly the sheep more than 20 years ago, a breakthrough that could boost medical research into human diseases. The two long-tailed macaques, Macaca fascicularis named Hua Hua and Zhong Zhong, were born at the Chinese Academy of Sciences Cass Institute of Neuroscience in Shanghai, and are the fruits of years of research into a cloning technique called somatic cell nuclear transfer. The barrier has been broken by this work, co-author Muming Pu, director of the Institute of Neuroscience of Cass Center for Excellence in Brain Science and Intelligence Technology, told AFP. Until now, the technique has been used to clone more than 20 different animal species, including dogs, pigs and cats, but primates have proven particularly difficult. The birth of the now 6- and 8-week-old macaque babies also raises ethical questions about how close scientists have come to one day cloning humans. Humans could be cloned by this technique, in principle, said Pu, though this team's focus was on cloning for medical research. In this January 22, 2018, photo released by China's Xinhua news agency, a nurse holds cloned macaques Zhong Zhong and Hua Hua at the non-human primate research facility of the Chinese Academy of Sciences. One day, the approach might be used to create large populations of genetically identical monkeys that could be used for medical research, and avoid taking monkeys from the wild. In the United States alone they are importing 30,000 to 40,000 monkeys each year by drug companies, said Pu. Their genetic backgrounds are all variable, they are not identical, so you need a large number of monkeys. For ethical reasons, I think having cloned monkey will greatly reduce the number of monkeys used for drug tests. Monkeys are commonly used in medical research on brain diseases like Parkinson's, cancer, immune and metabolic disorders. Much failure before success. The method used for these experiments is similar to that used to clone Dolly in 1996, but with several updates, said William Ritchie, an embryologist on the team that cloned Dolly the sheep at the Roslin Institute of the University of Edinburgh. The process involves removing the nucleus from a healthy egg, and replacing it with another nucleus from another type of body cell. The clone becomes the same as the creature that donated the replacement nucleus. We tried several different methods, but only one worked, said senior author Qiang Sun, director of the Non-Human Primate Research Facility at the Chinese Academy of Sciences Institute of Neurosciences. There was much failure before we found a way to successfully clone a monkey. In this January 22, 2018, photo released by China's Xinhua news agency, cloned macaques Zhong Zhong and Hua Hua sit in a lab at the Non-Human Primate Research Facility of the Chinese Academy of Sciences. Adult donor cells were attempted, but those clones died within hours of birth. What worked as replacement nuclei were cells that came from fetal connective tissue. Pu said it took first author Zhen Lu, a postdoctoral fellow, 3 years to perfect the procedure. The SCNT procedure is rather delicate, so the faster you do it, the less damage to the egg you have, and Dr. Lu has a green thumb for doing this. Ethical questions. Other monkeys have been cloned in the past by a different and simpler technique called embryo splitting, which mimics how twins arise naturally. The first primate ever cloned this way was Tetra, a rhesus monkey born in 1999. Embryo splitting can produce a maximum of 4 at a time, while the new technique could in theory clone far more. Still, the process that produced Hua Hua and Zhong Zhong remains very inefficient and hazardous, because the two babies were the only born from a group of 79 cloned embryos, said British scientists Robin Lovell-Badge, group leader of the Francis Crick Institute. While they succeeded in obtaining cloned macaques, the numbers are too low to make many conclusions, said Lovell-Badge, who was not involved in the study. With only 2 produced, it would have been far simpler to just split a normal early embryo into 2 to obtain identical twins, nor did the findings published in the U.S. journal Cell bring scientists any closer to human cloning. Lovell-Badge argued, this clearly remains a very foolish thing to attempt. It would be far too inefficient, far too unsafe, and it is also pointless. Darren Griffin, professor of genetics at the University of Kent, greeted the paper with cautious optimism, and called it very impressive. From a technical standpoint, the first report of cloning of the non-human primate will undoubtedly raise a series of ethical concerns, with critics evoking the slippery slope argument of this being one step closer to human cloning. He added, the benefits of this approach, however, are clear. A primate model that can be generated with a known and uniform genetic background would undoubtedly be very useful in the study, understanding and ultimately treatment of human diseases, especially those with a genetic element.